With that, I'd like to introduce our first speaker, Dr. Andrea Love from Nexcelom Biosciences, and she'll be presenting on functional in vitro assays for therapeutic antibody production and characterization. Andrea?
Thanks, Tina, for the introduction. I'm going to go ahead and share my screen. All right, can you guys see the PowerPoint now?
You're all good.
Excellent. All right, so thank you for having me. I'm going to present a little bit of data using image-based screening to utilize for screening and development for therapeutic antibody production. This is kind of the overview of my talk for today. I'm going to focus mostly on the applications themselves, but I'm going to quickly touch base on some of the challenges in the antibody discovery workflow. And this is true for therapeutic antibody generation as well as research antibodies. So obviously monoclonal antibodies are very useful tools. These were first generated using hybridoma technology over 4 decades ago. Monoclonal antibodies are used extensively across a broad array of different research areas, including things like immunodiagnostics, environmental monitoring, as well as basic science research tools. Antibody screening is, of course, important for discovering potential therapeutic modalities. As we've seen right now, there is a very heavy push toward developing therapeutic monoclonal antibodies for treatment of COVID-19, as well as using them as potential cancer therapeutics, other infectious disease therapeutics, and of course, autoimmune disorders as well. There's a variety of different methods in order to generate and screen antibodies. The traditional methods are using things like hybridomas, where you generate hybridoma cells by fusing myeloma cells with splenocytes. And then you're going to screen for antigen-specific hybridoma clones. There's other methodologies such as phage display or other sorts of mammalian or yeast cell display features as well. I'm not going to talk too much about that, but just really want to focus on the complex and time-intensive process in order to generate therapeutic or research antibodies. So as I mentioned, there are multiple strategies employed to do this. I'm going to focus really on the workflow for hybridoma technology, but of course, a lot of these assays and methodologies are relevant for the other workflows. The big takeaway here is that the workflow requires multiple screening steps from your cell line development process for isolation and evaluation of monoclonality of those samples, for actual monitoring of proliferation. So are these hybridomas actually growing? Once they start producing antibodies, are those antibodies actually binding the antigen of interest? And then once you've evaluated that they are binding, are they actually exerting the physiological effect of those? And at each of these stages, cell-based assays are critical. They're very unique. So a lot of times, multiple types of assays or multiple types of platforms have to be utilized for that. So I want to focus a little bit on some of the traditional methods that are used to screen for different stages of that workflow. Of course, we have flow cytometry, which is going to give you object level information by running your samples through fluidic sheath and generating scatter plot or histogram-based data. Of course, we have fluorescent microscopy, and that includes your inverted as well as confocal and other types of microscopes. Plate readers, and this could be luminescence or fluorescence, depending on which stage you're looking at. And then, of course, live cell imaging. Now, all of these have some limitations as well as certainly advantages, but I want to talk about an all-in-one solution that can enable you to really utilize throughout the entire workflow. So quickly about Nexcelom, we make imaging systems. The company's been around for over 15 years, and we specialize in image platforms for cell-based analysis. And this is for things as simple as automated cell counting for both cell lines and primary samples, to high throughput based cell analysis for viability, all the way up to plate-based screening. We have 4 global offices, and all of our field scientists are biology experts. And so we ascribe to the principle of fit for purpose, which enable us to collaborate and co-publish across a variety of research areas so we can spread best practices to other labs. So image cytometry is slightly different than flow cytometry and slightly different from fluorescent microscopy in that it merges some of those components to quantify images to actually produce cell level data. So it enables you to collect fluorescent-based and bright field images using fluorescent microscopy principles, but then analyze those in a manner that you would with flow cytometry data. So you can generate things like kinetic assays and also cell level data as opposed to well level data. Here's an example here of HeLa cells that are GFP transfected. And so you're actually able to quantify these kinetically without having to trypsinize those samples and run them on flow cytometry, but you're able to actually get parameters such as percent GFP positive, fluorescent intensity values on a cell level basis, etc. So it enables you to run these sorts of assays without having to harvest and trypsinize adherent cells or 3D cells, or even sacrifice suspension cells that you might want to maintain in culture. So Celigo is a plate-based high-throughput imager that utilizes a proprietary imaging platform that enables you to do rapid whole well imaging. It's equipped with 5 imaging channels, bright field and 4 fluorescent colors. You can see the optical configurations here. You can multiplex all of those into a single acquisition, so you can utilize it for a very diverse and versatile array of different assays. It supports all plate formats from a 6-well plate to a 1536-well plate. It can do whole well imaging even in those large well formats. And it's able to acquire these images in a very rapid fashion, as you can see in the video here. Approximate time to scan a 96-well plate in bright field takes about 3.5 minutes. When you add a second color, it takes 5 to 6 minutes, and so on and so forth for all 5 channels. We also have adapters for T25 and T75 flasks, 10 centimeter Petri dishes, and glass histology and chamber slides as well. So using this proprietary set of optics, which is actually an F-theta lens and a platform of galvanometer mirrors, you're able to move those light beams around the edge of the well and capture these very flat illuminated well images. As you can see here, this is very distinct to that of a conventional microscope, where you get often that donut illumination and you lose edge contrast at the edge of that well. This is critical for cells that often like to congregate along the well edge, because it'll enable you to sample your entire population of interest as opposed to just a few representative fields of view. So here is a very short video showing that stitching and scanning. So again, it will do this for every single well. For a 96-well plate, it moves a single position per well, and it will capture 16 images that it will auto stitch into a comprehensive whole well image. So the versatility of Celigo enables you to do a broad variety of assays across an entire day. Because it doesn't require you to leave the plate in the instrument, you're able to image and acquire a significant amount of data. This is just a very quick cartoon overview of how you can integrate it into your daily workflow, where you can utilize it for a broad variety of different types of assays. So now I'm going to focus on the applications themselves that come into play in the context of therapeutic antibody discovery. So the first thing is that you can utilize Celigo to establish and maintain cell lines. And this can be true for things like CHO cells, as well as other cell types such as hybridoma. So you can utilize it to monitor workflow steps such as your initial proliferation and outgrowth, transfection with traceable phenotypes, where you can monitor transfection efficiency, select cell cultures that have stabilized outgrowth. And then, of course, when you're isolating for monoclonality, you can actually detect single cell per well to verify before you monitor your colony that those cells, those colonies actually came from a single progenitor and not a potential doublet. So you can utilize our in-software gating interface to monitor fluorescent intensity as well. So you can use this to optimize transfection parameters or transduction parameters. You can utilize this to monitor proliferation rates amongst different cell culture in time to identify potential cell cultures of interest that are going to be better proliferative or expressing higher levels of particular antibody in question. So you can see here, even at low transfection rates here, we have a transfection rate of 0.7%. You're able to score those objects and identify them very easily. You have your GFP images there, your bright field with the GFP merge, and then the object segmentation is denoting the cells that were identified as being GFP positive in the red color coding versus your total population in the blue color coding. You can use this kinetically, so you can monitor outgrowth. So this was a single cell sorting experiment where you have your bright field with your GFP image. You can verify that that is in fact being counted as a single object of interest. And then you can monitor expansion of that cell over time. So by day 2, you have 4 cells, and by day 3, you have 8 cells, and so on and so forth. So this enables you to monitor this without having to wait several weeks for your colony to grow. So here's just an example of utilizing this from a whole well point of view. So by screening these cells, you can actually visualize single cells, even if they're at the very edge of the well, which we know cells like to do. And then you can monitor that single clone expansion all the way out. So after you do your single cell sorting or your limiting dilution, you can actually use this to score your wells of interest. So here you have, your goal is to plate 1 cell per well. At day 0, you can identify that. We have 1 well that has a single cell, 1 well that has 2 cells. Now, ultimately, they're very close to each other in proximity. So if you weren't scoring this at the time of deposition of those cells, you might actually lose that information. Now, in this instance, you can see these are hybridoma cells that you have a single distinct colony at day 10 with your 1 cell per well, 2 colonies that are very distinct there. So this enables you to sacrifice and exclude that well from future scale-up culture. Now, of course, you can do this at the level of bright field only, but you can also use this with fluorescence. And often we recommend using a fluorescent cell tracker dye, or so often we're using transfected cells that are already expressing a fluorescent reporter, which enables us to score this even more accurately. So by using our heat map function, you can score these objects at the level of cell sorting, but then you can also take that a step further and score that after you've allowed those cells to proliferate. So a lot of times, even though you may have something like 85% occupancy after a sort, most of those wells may not in fact proliferate due to trauma to the cells or other parameters in there. So this enables you to multiplex your single cell detection with your single colony verification, so you can identify cells that not only had a single cell at the time of sort, but also proliferated. So now, once you've got those cell lines established, you want to actually determine whether or not they're producing antibody of interest and whether or not that antibody is actually binding and specific for the antigen of interest. So the traditional screening methods for antibody binding are typically using either ELISA-based or flow-based methodologies. Now, ELISA, of course, you have limitations where if you have your antigen as surface marker expressed, sometimes that can be challenging to actually get your ELISA plate coded with the proper configuration of that protein. Oftentimes using high throughput screening with a brute force approach on ELISA can yield false positives or false negative results. Using flow cytometry, of course, is very sensitive, but it's very time consuming. So kind of scaling up that methodology in a similar approach on a plate enables you to test multiple parameters in a bit more rapid fashion. So Celigo enables you to do intact functional cell-based assays using high throughput screening. So you can do something like a 96 well plate in about 9 minutes. So the data here is looking for binding of hybridoma supernatant. So here we have CHO cells that are expressing either the antigen of interest, in this case this is CD39, or wild type CHO that are not expressing that. These are differentially labeled. So we have CFSE labeled cells that are a wild type versus the antibody of interest, which is Alexafor 594 labeled. We labeled, this is actually in a co-culture scenario, so you don't need different experimental plates. You can actually do this in the same plate to measure specificity of your binding. So all of your cells here are labeled with hooks, so we're monitoring the sensitivity and specificity of the antibody against CD39 here. So then, of course, we monitor the scatter plot interface, and this is actually exported to FCS Express software. Celigo, all of that cell level data is also compatible with flow cytometry software packages as well. So what you can see in the scatter plot here is that your hybridoma supernatant is eliciting binding on your antigen of interest expressing CHO, but not on the off-target wild type CHO. And you can very easily see that distinct population split in your scatter plot there. So you can utilize that to monitor binding efficiency. So here you have differential binding in different potential target antigen or antibodies rather. And you can see that the data that we captured on the image cytometer is very comparable to the flow cytometer. You have higher affinity with certain antibody and then low binding with other antibody tests. Using this approach, you can identify hits. So here we screened 576 supernames across 696 wall plates, and we identified 16 positive hits. And those are color-coded in that heat map function here. These values depicted are the KD values. So we did binding coefficient analysis using the image data acquired. And this enables you to be able to co-culture, reduce the number of reagents utilized for screening, and capture this in about an hour and a half versus upwards of a day or more. So here's looking at just some of our binding affinity curves. So you obviously have your appropriate controls in here. You have your different test antibodies as well as some purified antibody, the relative concentrations in micrograms per mil. You can see by eye that you have different differential intensity just by eye. And then of course you can export that raw data to GraphPad and then calculate your binding coefficients. So another functional antibody assay you can employ is antibody internalization. So we use pH roto here as a probe, which is obviously going to fluoresce red in the acidified lysosome of a cell that has internalized that antibody of interest. We have a few different test cells here to monitor uptake. So we've scored these potential targets based on positive or negative uptake. You can see some images here with the different antibody that we tested. You can see that you have some that have high amounts of antibody internalization, such as the compound SYD977 that was taken up by the SKBR3 cells. And then, of course, you can see the data plotted here as a function of the proportion of cells in that culture that have actually taken up the antibody of interest. You can also plot that as a function of fluorescent intensity, like we did with the antibody binding curves. So once you've established that you can actually quantify binding affinity of your antibodies that you're producing, you want to evaluate whether or not those antibodies actually elicit the physiological effect. So functional assays are very broad and diverse. Obviously, in the context of viral infections, we're looking for antibody neutralization, where we're actually blocking viral infectivity. For things like cancer immunotherapy, we want to be sure that those antibody-dependent treatments are actually eliciting cytotoxicity of your cancer cells or eliciting phagocytosis by, say, macrophages. And then, of course, monoclonal antibody therapies, where you're actually inhibiting a pathway step and directly eliciting apoptosis of cancer cells or similar. So here's just the general workflow for an antibody neutralization assay. Of course, you have your functional antibodies that are produced by binding to, say, a virus of interest. You're going to interfere with the binding of the receptor. You might lead to things like opsonization if it's a bacterial pathogen, but ultimately you're going to inhibit the infection of the actual virus of interest. You can utilize this antibody neutralization approach in suspension cells and adherent cells on the Celigo platform. So here we have some examples of Zika infection, and you have Raji cells as well as Vero cells, so two different cell culture models. Bright field here or hooks are identifying your total cells of interest, and these are actually GFP labeled Zika virus particles, so all of your green cells are infected cells. So utilizing this approach with different concentrations of various test antibodies, you can actually employ antibody neutralization right on the plate in a non-destructive manner, and you can actually calculate your IC50 curves. Now, we did validate this to flow cytometry, and the values were quite comparable across the board. You can also employ antibody screening for ADCC. So this is antibody-dependent cell cytotoxicity, where you have an antibody that's going to bind an antigen on a target cell. In this case, this is a cancer cell antigen. That's going to recruit NK cells that are going to bind CD16. That's going to elicit degranulation and, of course, apoptosis. We use this by directly labeling our target cells using various cell tracker dyes such as calcium AM or CMFDA. And then you can actually quantify the cell death kinetically as a function of loss of cell fluorescence over time. So utilizing this approach, again, you can screen a variety of different test antibodies, both in a dose-dependent manner in the first figure on the bottom left, or as a time-dependent manner, as you see in the bottom right figure. And then, of course, the images there, you can see over time a distinct loss of number of green cells, which indicates a loss of viability. Taking a similar tack, you can also monitor things like antibody-dependent cell phagocytosis. And so this is utilizing a similar approach, except instead of cytotoxicity, you're recruiting a phagocyte such as a macrophage to actually bind to and phagocytose the target cell. So here what we have is your target cell is labeled with CFSE, your macrophage is labeled with cell trace violet in this instance. You can see your bright field and your fluorescent merge. So you're looking for that co-localization. You can also use other sorts of reporters like a pH roto to monitor acidification of, say, the lysosome. And then, of course, utilizing the scatter plot interface to score the proportion of objects that have elicited that ADCP effect. And then the last example that I'm going to quickly touch on is utilizing Celigo to screen for monoclonal antibody therapeutics. So here is an example of a PD-L1 inhibitor, which is atezolizumab. And this is used on Celigo to actually screen for synergistic effects to elicit cell cytotoxicity in triple-negative breast cancer cells. And so here was a screen done. This is actually published data in oncoimmunology, where we screened the ability of T cells to elicit cell killing with atezolizumab treatment, as well as augmenting it with either a proteasome inhibitor or lysosome inhibitor as well. And you can see in the figures there that utilizing combinatorial therapy actually augmented the ability of the monoclonal antibody therapy to elicit cell killing. Again, this is just utilized in a kinetic manner by screening these cells over time on the Celigo. So to summarize very briefly, Celigo can be really utilized for the entire antibody generation workflow, from establishment of your cell lines, monitoring outgrowth of those clones to colonies, to actually monitoring the production and binding of those therapeutic antibodies that are produced by your hybridoma cells, and then of course utilizing for functional assays. The advantages of using image cytometry is going to enable you to increase your screening speed, increase the throughput, enable you to quickly subclone and expand, something that might take 6 weeks or longer. You can actually speed that workflow up significantly. Because you're able to co-culture and utilize multiple parameters on the same plate, you can assess more experimental conditions than traditional historical methods. And then you can multiplex actually assess both production and function of the antibodies that you're generating. So this enables you to do kinetic and real-time assays, as well as endpoint-based assays to serve as an all-in-one solution. So that's my overview. I'd be happy to take any questions.
Thank you so much, Andrea. That was wonderful. So we have one question from Kim Cusser. She wanted to ask, what is the learning curve for the Celigo software?
That is a great question. So I would say the learning curve is certainly less steep than learning flow cytometry. And I would say compared to other plate-based imaging system, it's very user-friendly. The software has only a few key parameters that you really need to become very competent with in order to image and analyze. When we do training, we focus on image acquisition and then image analysis as two distinct topics. I would say within a 2-day training, people are pretty comfortable running those assays on their own. We do also provide ongoing training and support as your experimental design expands and gets more complicated.
Great. We have one comment from Ben Daniel. He said he can't wait to get his installed. He's super excited.
We are also very excited, Ben. We can't wait to get back in the lab with you.
And then Su Hong Ho has a question. Is the Nexcelom instrument also good for suspension cells?
That's a great question. And the answer is absolutely. So suspension cells, because they're very round and bright, they actually image quite beautifully. We have recommendations for actually plating those cells and imaging, but we utilize those very routinely. I've been working extensively with things like NALM6s and K562s for a lot of CAR-T based screening right now.
Wonderful. And then from Thomas Miller, what and how many fluorescent parameters can be measured?
That's a great question. So you have 5 different colors that you can work with. So you can identify things like number of objects, fluorescent intensity on a per object basis, or normalized to size of the cell. So we use both a mean intensity or fluorescent intensity. You also get parameters such as size, XY position, co-localization. So you can do a lot of the gating interfaces that you would apply with flow, where you're looking for, say, double positives or single positives or things like that as well.
Okay, great. And then just personally, I know we've had a bit of experience with this. Another question popped through though, so I'll hold my question. So Svetlana Mazel is asking, how sensitive is the Celigo for the single cell deposition?
That's a great question. So efficiency, if you're using a fluorescent tracker dye, such as a calcium AM or a CMFDA, or if your cells are already fluorescent protein expressed, is 99% or greater. If you're using bright field only, you can't exclude things like media artifacts and artifacts on the plastics themselves. So that decreases a little bit to kind of the mid 80% efficiency. And that's typically why we recommend using a non-toxic cell tracker dye. If you take the bright field approach, we simply will screen those plates, over sequential time points to verify that, say, an object scored in bright field is in fact a cell that's proliferating.
Wonderful. And then Andy Morshauser asked, are single color controls necessary for compensation?
That's a great question. Now, typically no, and that's because we're using LED-based light sources that have paired excitation emission, and they're only illuminating independently. We also do have a software-based background correction that excludes a lot of that potential spectral overlap. Because you have only 4 different sources of light, you have a lot less of that than you would encounter with flow. We do have some people that will include those just as an internal control, so you can verify, background autofluorescence and whatnot. But generally, it's not actually required for your analysis.
Great. And then personally, I just wanted to ask, thank you again for this wonderful talk. I wanted to ask about the PCR plates and, any recommendations on PCR plates and what to take into consideration with those.
Yes, so there is a single manufacturer of flat bottom PCR plates that I found. I believe they are semi-skirted, 96 well, flat bottom PCR plates. They image very beautifully on Celigo. They have a very, very nice flat bottom. They're great for imaging. So you can actually use those for single cell sorting directly into PCR plates, where you can actually image and then score your single cells before doing your sequencing. Traditional PCR plates, because they're round on the bottom, they have a lot of optical interference, and so they're not great at imaging. So if there was an application where you wanted to utilize it for direct sorting into PCR plates, there is a plate option that we've tested and validated, and it works very nicely. If you're sorting single nuclei, you don't want to sort into lysis buffer because that will obviously disaggregate any of your DNA and you won't be able to identify your nuclei. So ultimately, you want to sort into, say, a buffer or a media, and then add your lysis buffer after confirming which wells have single nuclei.
Perfect. Thank you. All right, that's actually perfectly on time. And then that's all the questions that we have. So it worked out timing very nicely. So thank you so much, Andrea, for a wonderful presentation.
Thank you. And feel free to email if anybody has any additional questions you think of afterwards. My email's right there on the screen.